Hello, students. We can't wait to work with you in the lab in the next couple of weeks. In these short videos, you're going to see what we're going to be doing in the lab. You'll get to see the faculty going through the procedures. This will afford you the opportunity to see the materials we'll use in the lab, to think about the sequence of it, and also to come to lab prepared with some questions if any are raised. Uh, so this first video will cover Lab 1, and there will be 2 subsequent videos that cover Lab 2 and 3. The very first thing we will do is make a hand stamp on an agar plate to see what microbes exist on our hands. Whenever you're working with plates in the lab, you're going to label them. You're going to label them on the bottom so that the lid can't get separated from the important part, which is the agar media and the microbes and plaques that you put on it. The thing to do here is to put your identifying information for your group, the date, and then anything else that your lab fellows might tell you to write. So here we see 30 degrees Celsius. And also notice that this labeling is really in the corner and it's very small so that it doesn't obscure our view, because what we will be doing is looking for microbes on these plates. So you're going to label the plate here. You're going to turn it over. And the very first thing we're going to do is just test out the gelatinous structure of the agar we're going to be using. So we just gently press our fingers on a test one. So we don't push our fingers all the way through to the bottom or, make any other kind of aggressive move that would affect the media. Once we have these out of the way, they will be taken and put away for a couple of days till we get back into the lab. This time delay will afford an opportunity for the microbes to grow. The agar that is inside the plate is a growth medium, so it's the niche, it's the food, shelter, and water for the microbes. This is an intense dramatization of what we might see. We will not see something like this because we don't have enough time to grow our microbes on our media, but I foreshadow here to give you the sense that we're going to use visual cues to identify microbes. Now, we're going to start following along with the lab manual. So on page 1, quadrant streaking. So we're going to take one colony, we're going to select one of those colonies from our hands using a wooden dowel, and you're going to grow that out. So we need to get down to a place where we can see an individual colony of microbes so we can identify them. So you're going to select one of your colonies on the handprint that grew well. You'll label the LB agar plate with your initials, the date, and where the isolate came from. So the isolate is just one microbial blotch that you pull off of your handprint. You'll take a sterile wooden stick and gently scoop up the small quantity of the isolate from your plate. The sticks should not be thrown into the biohazard waste. Collect them in the used stick receptacle that's on your table, and those will be sterilized. So you're going to use this pattern. You can see it here in these plates. This is called quadrant streaking because you're going to visually separate the plate into 4 quadrants. You're going to use the stick to gently streak the bacteria over the new plate in the pattern shown here. Essentially, you're putting the isolate down in one quadrant and then you're crossing. Just use the stick to cross through that just ever so slightly, make a few lines in the next quadrant, and then you cross into the next quadrant. So we go like this, and then the second one is like this, And then the third one is like this. And then finally at the end, one cross and come to the middle. Okay, that last one should involve traveling towards the middle of the plate. Each successively lighter streak should be done with a fresh sterile stick. Only the first stick is to be picked up bacteria from the other plate. The others are only to spread the bacteria you transferred with the first stick. Place the plate in the incubator overnight face down. Your lab fellow will do this for you. In the next video, we'll move on to the next lab.